Well, welcome everybody to the New Jersey Climate Change Resource Center's Summer Climate Academy's webinar on harmful algal blooms. I'm Marjorie Kaplan. I'm both Associate Director of the Rutgers Climate Institute and along with my colleague Jean Herb of the Blaustein School of Planning and Public Policy, I co-direct the New Jersey Climate Change Resource Center. A few logistics. Today's webinar is going to last until 1:30, and other than today's presenters and our Rutgers team, you're all muted. So to communicate with us, please type your questions into the chat box, which you'll find at the bottom of the screen. There's a little bubble there. It says chat, and you can send us questions and comments, and please send your notes to all panelists, as that ensures all of us working behind the scenes today will see your questions. Well, I want to say thank you to my colleagues, Matt Drews and Dr. Carrie Ferraro at Rutgers, and Jessica Spinagle from the New Jersey Department of Environmental Protection for their help with today's webinar. I also want to let you know we're recording our webinar and will be available at our website, which I can see Carrie's already put into the chat. There are 1.5 certification maintenance credits available through the American Planning Association, and 1 continuing education credits has been requested through the Association of State Floodplain Management. So reach out to us after the webinar if you need a certificate, or you can stick it in the chat box. We'll be able to check that. Today, we are very pleased to have 6 amazing speakers from the from the New Jersey Department of Environmental Protection, from Clean Waters Consulting, and the Watershed Institute joining us. In the interest of time, I'm not going to go over all of their bios, but we encourage you to check them out on our website. I'd like to remind everyone again to put your questions in the chat box to panelists. And to kick us off today, we're going to hear from Dr. Steven Souza from Clean Waters Consulting, who'll provide an overview of harmful algal blooms and their relationship to climate change. I'm going to turn it over to you, Steve.
Great, thank you, Marjorie. And next. And I want to extend my thanks to DEP and Rutgers for having me here, with this exceptional panel of experts on harmful algae blooms. Next. So unfortunately, the term harmful algae blooms has become part of our lexicon. It's in our day-to-day conversation, it seems, in the middle of the summer. And as we all know, these are intense blooms that are caused by cyanobacteria, species and organisms that in the past we used to refer to as blue-green algae. But cyanobacteria are bacteria. They're prokaryotes, not eukaryotes. However, they can photosynthesize, so they definitely share properties with both bacteria and algae. And for us, the problems are that when we have a bloom, that's defined by high concentrations of the organisms themselves, high cell counts, as well as cyanotoxins. Next. And these are what I call the bad guys. A lot of you have become familiar with the names of these various species. 2 of them that you may not think about that often are Lyngbya and Gliotrichia at the bottom of the list. These are benthic forms that actually grow at the bottom of lakes, ponds, and reservoirs at the sediment water interface. Next. So what makes cyanobacteria so problematic? Well, they really have evolved a whole bag of tricks that they utilize to give themselves a jump in the environment and outcompete the good algae. Number 1 is that many of them can assimilate atmospheric nitrogen, and this provides them with an unlimited source of that important metabolic building block. Secondly, they are biologically adept and efficient and effective at assimilating organic phosphorus. And the source of organic phosphorus is dead algae and weeds, and we'll come back to talk about the importance of that later. Many of them can regulate their position in the water column, allowing them to rise and fall over the course of the day, and either to take advantage of changing light conditions or even nutrient concentrations. Many of them, as I mentioned before, those benthic forms can do really well under low light conditions. And then another big thing is that they're rejected as food by filter feeders, including very prolific filter feeders such as zebra mussels and quagga mussels. For us, the problem is that they tend to bloom and become problematic in the middle of the summer when, peak recreational use of lakes and reservoirs and ponds. And the big problem also is that they produce toxins that definitely impact humans, pets, and livestock. Next. Cyanotoxins, I'll leave it to Vic Poretti later to talk more about cyanotoxins, but keep in mind that this is a defensive or competitive mechanism that they've evolved. That they didn't evolve producing cyanotoxins to impact humans, because they were around long before humans were. They really, produce cyanotoxins to really give them the ability to outcompete phytoplankton. And yes, these organotoxins impact the central nervous system of mammals, as well as liver and even skin. Endotoxins, they're released by both living and dead cells, but really high toxin levels are typically measured during the die-off of a bloom, whether that's something that has been facilitated by an algicide application or just by the natural die-off of the bloom, or when the cells become physically disrupted by wind, wave, wake action. Once these products are in the water column, they're extremely stable and very slow to photo or biodegrade. Next. So again, Vic will talk about more about cyanotoxins, but bottom line is that they will make you sick. Not only will they make you sick, but they'll make your pet sick as well. Next. And those of you that do have dogs like myself, when you see a bloom, you need to keep your pets out. And any of you that do want to learn more about how to protect your pets from harmful algae blooms, download any of these technical documents. Next. So some common perceptions about HABs. We've heard that it's something that only occurs in nutrient-rich water bodies, hypereutrophic water bodies, but we've come to know that these blooms occur even in what would be considered almost pristine mountain lakes. And the second thing is that we can deal with this by doing heavy treatments using some type of algicide, primarily copper sulfate. Next. However, as I'll show, that can actually make things worse. Next. So the paradox of copper sulfate treatments is, yes, there's nothing wrong with using copper sulfate or other algicides, and they should be part of your HAB toolbox. However, they can't be the go-to reaction, and these products have to be used carefully, proactively, and very selectively. Because what happens is that, yes, they'll knock down the bloom, they'll kill the cyanobacteria, and you'll get some temporary relief. But at the same time, what ends up happening is that when you kill off those cells and blow them up, and that's essentially what happens with an algicide, you release very rapidly large amounts of cyanotoxins in organic phosphorus. And remember, I mentioned previously that cyanobacteria are very adept at utilizing organic phosphorus. So what you've done now is basically created an environment that really facilitates more and more cyanobacteria growth. In addition to that, you've killed off the good algae, so there's no competition now for nutrients. You've killed off zooplankton. And if you release a lot of organic material into the water column at once, the bacterial decomposition of that can depress oxygen levels, and that leads to a fish kill. Next. Okay, so what are some of the typical conditions that favor a HAB? Yeah, as I mentioned before, it's what we see in the summer, very warm water temperatures, plenty of light for photosynthesis, very stable water column. But on top of that, the quick influx of phosphorus that originates either from internal or external sources. Next. Phosphorus is definitely the common denominator. The more phosphorus, the more productivity, and that leads to more cyanobacteria. And you don't need a lot. You only need about a pound of phosphorus to stimulate about 1,000 pounds of cyanobacteria growth, and concentrations as low as 0.04, which is less than the state water quality standard for total phosphorus, which is 0.05, is more than enough to stimulate and sustain a HAB. Next. So where is all this phosphorus coming from? Next. It comes from a variety of sources, both internal and external. And these vary seasonally and under various types of conditions that facilitate either more or less of the release of these nutrients. We can't overlook waterfowl. Next, keep in mind that 4 geese produce in one day the equivalent amount of phosphorus as 1 septic system. Next. The bottom line, though, is that the majority of all of these sources, whether internal or external, are definitely affected by climate change, both in terms of the amount that's being released and the frequency at which these nutrients, the phosphorus, is entering our lakes, ponds, and reservoirs. Next. That climate change link, there's more and more data that has been developed showing that there's a positive correlation with the frequency, intensity, and duration of HABs. And most of this has to do with warmer winters, as I'll note in the upcoming slide, as well as increasing amounts of phosphorus, as well as nitrogen, that is introduced during the growing season due to an increased frequency and intensity of storm events. And then warmer summers. So basically, altogether, that creates an environment that favors cyanobacteria growth and the development of HABs. Next. Basic takeaway from this slide, which is data that was developed by Dr. Anthony Broccoli of Rutgers, is this trend in warmer winters. And with warmer winters comes earlier ice out. You can go to the next slide. And with earlier ice out, you get an earlier onset of the growing season. And we saw this in 2019, where we had early ice out, a very warm winter. That on top of that, we had an exceptionally wet winter in spring. So there's a lot of external phosphorus loading, saturated soils which increase septic loading that Gabe will talk about. And that also decreased the opportunity for stormwater retention and infiltration. And then in July, we had these intermittent extreme rainfall events that led to more phosphorus, more water column mixing, and all of that promoted HAB formation. Next. So yes, there is a connection with climate change. We'll learn about that more today. And even though HABs are not a new thing, their occurrence, severity, and duration definitely increasing, and that is definitely linked to climate change. So with that, I thank you, and I'll remain available later on for questions.
That was fantastic, Steve. Thank you so much. That's just a wonderful presentation, wonderful overview. And so now I'm going to invite Vic Poretti from DEP, who will speak to how DEP does HAB monitoring and some results from the monitoring. So take it away, Vic.
Thanks, Marjorie. Hi, everyone. I'm Vic Poretti. I'm with the New Jersey DEP Bureau of Freshwater and Biological Monitoring, and we're responsible for responding to HABs in fresh waters, recreational waters, and drinking water sources. And I'm going to talk to you today about how exactly we do that and summarize the findings to date on our HAB response. Next slide, please. The DEP has a recreational response strategy that we developed in 2017. It's for recreational waters and sources of drinking waters. We have risk thresholds based on cell concentration and toxin concentration. The three toxins originally established for risk in the strategy are microcystins, which are the most common, cylindrospermopsin and anatoxin A. We added a fourth toxin, saxitoxin, this year to the 2021 strategy. So each year we review the most current available science, and based on that, the three existing toxin levels were slightly lowered. We also reviewed the cell count thresholds. They will remain the same for the 2021 strategy. The strategy was released this spring, and if you visit our website, it's available on our website. And I also would encourage anybody to, we have a listserv registration for any HAB news. So I would encourage anybody to visit our website and register for that. Next slide, please. So this slide shows the old and new thresholds for toxins. So MCT, microcystin, cylindrospermopsin, anatoxin, and saxitoxin. The important takeaway here is, so the levels were slightly lowered for the three existing to 2, 5, and 15 respectively. The important takeaway is for at least Cylindrus, Bmps, and anatoxin, we had no detections above these thresholds with the current thresholds or the previous thresholds. So we don't really get a lot of detection on those two toxins. For microcystins, revisiting previous data, out of 900 samples, there were about 69 sites that the microcystin levels were between the new threshold of 2 and the old threshold of 3. All but 7 of these would have had an alert posted based on cell count alone. So a very low amount. For saxitoxin, again, we looked at past data. There was only 2 cases that saxitoxin was above the threshold of 0.6, but in each of these cases, advisories would have been posted for cell counts and microcystins regardless. Next slide. We reanalyzed our cell concentration versus potential for toxin production at the new microcystin level of 2 micrograms per liter, using all data that we had to date. And this graph shows that the threshold of 80,000 cells per mil is still the point where we see that toxin production becomes more significant even at the new threshold of 2 micrograms per liter. So this 80,000 threshold is where we advise for beach closings. So that's an important number there. And that number hasn't changed with the new 2021 strategy. Next slide, please. So, how do we apply our laboratory results? In 2020, we enhanced our communication system. We now have a tiered alert approach to protect recreation and water and to communicate what people can't do and what they can do with caution based on cell concentration and toxin concentration. So I just want to note that DEP doesn't close lakes or beaches. Beaches are closed by local health authorities under the New Jersey Department of Health sanitary regulations. Those closings are based on results we provide and recommend, but our Bureau only tests for HABs and recommends beach closing based on HAB-related data. So beaches are required to be monitored for bacteria on a weekly basis. So there may be instances where a beach is closed for high bacteria counts, not necessarily a HAB, or maybe some other reason the beach is closed. There might be debris in the water and such. So our findings are only HAB related. So, if you come across the beach closing and you don't see a HAB there, it may be for another reason. Next slide, please. So to communicate our information, we have a HAB interactive mapping and communication system. It's used to both report suspected HABs by the public and display data and alerts. The reporting system and map, they've been extremely successful and widely used by the public. The report a HAB submission form can be accessed directly from a link on the MAP website. There's also links on our various HAB web pages that you can access to report a HAB submission form. It can be used with a smartphone or a PC, and it provides a lot of information for us. It pinpoints the site. You could upload photos. You could record observation and field data. And the good thing about it is we our HAB response staff get an immediate email, and that allows us to have a faster and more precise response. Next slide, please. So once we sample and have laboratory analysis performed, data and alerts are uploaded to the dashboard as soon as they're available. In the lower left-hand corner, there's various links. One is a tutorial that would walk you through the various functions of the dashboard and how to use it. The dashboard right now has only 2021 sites, and you can access data from 2020 and previous years using our new data download function. Again, there's a link on the bottom left-hand corner of the dashboard. Next slide, please. So once you get to our data download site, it has similar functions. You could search for sites and click on the dots and get data, but you could also download any data we have to date. You could do it by water body, you could do it by site, you could download every piece of data we have. This data becomes available immediately after it's submitted to our dashboard. Next slide, please. Some other monitoring we do. We're expanding our continuous real-time buoy program. The buoys provide information on phycocyanin, which is a pigment unique to cyanobacteria, and we can approximate cell counts using phycocyanin measurements. The buoys also provide water quality data such as pH, temperature, conductivity. This allows us to characterize what's going on in the water body and possibly predict when a HAB might occur. When you go on the website, this is you could download a graph similar to this. The blue is the phycocyanin pigment. The black here, if you chose, it's pH, and you could see there's some pH spikes that precede the phycocyanin spike. So pH rises are kind of an indicator in some water bodies that you might get a HAB bloom soon. So it's a lot of good information from these buoys. And again, it's real-time data. I just like to, caution people, when you see this information, we use this information to monitor trends, what's going on, and this alerts us to deploy staff for sampling. It doesn't replace lab analysis or lab confirmation analysis that are used for the alerts on the map. Next slide. We also have a flight reconnaissance program. Again, this uses a hyperspectral sensor to monitor phycocyanin. And again, we convert that into cell count approximations. These are done at larger lakes throughout the state. Again, we use this to inform us of where we need to deploy staff for sampling and monitor conditions of existing HABs. So for instance, these flights, the dark gray black is very low counts, the green is moderate. If we see the changes that go from like an orange to green, we know that things are getting worse. We have to deploy staff to do sampling and lab confirmation analysis. Next slide, please. We also use handheld phycocyanometers. Again, this is phycocyanin. It's a real easy measurement to do. These are inexpensive, and we have a loan program through our bureau now that we loan to partners. These could be used to surveil HABs, screen for suspected HABs. If there is a confirmed HAB, it can be used to monitor status. So when the status goes up and down, again, we could deploy staff to sample. I just want to note, using phycocyanin measurements, they do approximate cell counts, but they don't approximate toxin levels. So that's why it's very important to do confirmation lab analysis to confirm what's really going on in the water body. Next slide. So lastly, I'll just give a quick summary of the 2020 season and information to date. In 2020, we had an increase in HABs reported rising from 74 to 83 water bodies, and confirmed HABs rising from 35 to 47 water bodies. HABs confirmed the drinking water sources doubled although overall it's a very small amount. They went from 4 water bodies to 8. Not a real significant amount of drinking water sources had HABs. HABs at beaches decreased slightly, but again, they went from 6 to 4. There's about 360 freshwater beaches throughout the state. So 4 closures is a very small number. Next slide, please. Back one. There you go. Thanks. We have a 2020 annual summary report available at this website. Gives a detailed information on the 2020 season. We had an 11% increase in reports of suspected HABs. We're often asked if the increase in reports is due to more awareness by the public or actually more occurrence of HABs throughout the state. Based on our data, I would say it's the latter. We had a disproportionately more confirmed HABs, 26% increase in confirmed HABs at the watch or alert above levels. 54% of water bodies with confirmed HABs in 2020 had confirmed HABs in previous years. So if the water body does get a HAB, it tends to repeat year after year. And we also had a 47% increase in HABs persisting into and past December. So there's a good portion of water bodies that did have confirmed HABs. They continue to linger throughout the winter. Next slide is here's my contact information. Please feel free to email me with any questions. I'd be happy to answer them, and I'll be around at the end to answer any questions you have. Thank you.
Thank you so much, Vic. That was really, really interesting. I'm going to now invite Rob Newby from DEP, who going to speak to his work on how species and toxins move through the watershed. So Rob, take it away.
Thank you so much. All right, next slide. I'll give you a real brief overview of kind of the impacts that climate change is having on cyanobacteria, just as a real kind of overview as to why we are looking at something as, kind of widespread as toxins moving through a watershed. But real kind of brief, we know that climate change and cyanobacteria go hand in hand. So about 2.4 billion years ago, the earliest cyanobacteria evolved in the oceans, and they're actually what's responsible for oxygenating our atmosphere. And the conditions were just perfect for this to happen. Combination of high temperatures, combination of right amount of gases, just made it absolutely perfect for them to evolve, to start photosynthesis and start producing oxygen. And cyanobacteria are pretty ubiquitously found throughout ecosystems. You can find them in soils, you can find them in freshwater, saltwater, estuaries. They're pretty much found throughout the entirety of the planet. And we do know from both empirical and from observational evidence that nuisance problems caused by cyanobacteria called blooms are increasing in frequency, and they're getting progressively worse. Next slide. So what we're seeing basically is that when you model data and you start looking at some real world data, that you're getting that the problems caused by cyanobacteria are increasing both spatially and in magnitude. So we're getting longer blooms that are occurring over wider areas. Next. We can use Lake Erie as an example. Everyone has heard of the problem that happened with Toledo, Ohio, which shut down the use of their water or for drinking and for use. But this problem on Lake Erie is going back, decades, right? So starting in 1984, and this is from Landsat. So we can see that looking at this data from collection of just, this is looking at chlorophyll, you can see that the cyanobacteria, the bloom, has basically getting, it's gotten worse. And there are some years where it's not bad, but then you can see that 2010, or starting really around 2006, it just starts to, expand in magnitude across the entirety of the lake. And there's a number of reasons why this is happening, and temperature is a likely driver, or likely key driver for this. Next slide. So when we're looking at some other key features in terms of, like modeling data based on what we understand so far, based on key drivers, we are expecting surface water temperatures to rise at least 0.5 degrees. That's the very minimum of what we're expecting. So depending on how you do your modeling, it's either 0.5 to 2, or it could be as high as 2 to 5. Next slide. This kind of shows you for the United States what that looks like. Obviously, we are kind of in a weird kind of area for New Jersey. If we are in the HAGM, the bottom one, we are going to be greatly impacted by such an increase. And this is looking basically over the next 50 or so years, what a potential impact could possibly be. And how this relates to cyanobacteria, next slide, is that basically this is going to change the assemblages. So Steve had mentioned before that as you increase temperatures and as you start to see these changes in sort of winter weather patterns, that it causes cyanobacteria to come out earlier, changes the way that green algae and diatoms tend to interact. And we know that because cyanobacteria are pretty hardy, that they tend to get a foothold very early. Next slide. And what that looks like is basically that we're looking at either two types of scenarios. One is a linear growth scenario, one is a plateau growth scenario. And they're pretty much both likely scenarios given how we are projecting forward with our current path. But what we're kind of expecting to see is that we're expecting to see a larger increase of frequency of cyanobacteria growth in many of our surface water bodies. New Jersey is going to be impacted. You can kind of see no matter what, whether it's plateau or linear, by 2050, we are expecting to see at least a 2,000 cell per mil increase. On the low end, and on the higher end, we're expecting to see possibly at least a 6,000 cell per mil increase. Next slide. So what we're seeing now, basically, this is data collected from USGS using kind of just a wide scale surveillance, is that we're seeing blooms that are spread throughout the entirety of the continent. There are areas where it's relatively low in frequency, but it is fairly wide. And there are areas where there's an alarming concentrations of cells per mil. Next slide. When we look at toxins and we look at basically as things sort of move through, because we know that many large water bodies drain to river systems, we are seeing that toxins do permeate into our river systems. And alarmingly, what we're also seeing is we're seeing not only toxins, but we're seeing that there are cells, and we're seeing that there are the genes that can make the toxins that are present in these river systems. So look at the Delaware River, they found this gene to control the production for microcystin and for spermopsin, but they did not find the toxin. So what's important to notice is that we don't fully understand the entire process of how toxins are produced, but finding the genes is kind of almost like a smoking gun. We know that there's the potential now that the toxins could eventually be produced. Next slide. So what's sort of unknown currently, again, it's, we have some knowns. We know that climate change is a big driver, both directly because temperature impacts the cells. The warmer the water, the warmer the surface temperature of the water, the more likely it is that you're going to get planktonic blooms of cyanobacteria. And we've seen this, based on what we have for measurements, and we've seen this globally, we've seen this internationally. It's no surprise that the warmer the water, the more likely it is that there's going to be a bloom. And we also know that the warmer the water, it's a tongue twister. The warmer the water gets quicker, the more likely it could possibly be toxigenic. But there's also unknown factors with this. We know that water conditions play a role in the dominance of toxigenic versus non-toxigenic cyanobacteria, but we don't fully understand this process. We think that some of the toxins they've evolved to provide oxidative stress relief for the cyanobacteria, but we don't fully know yet. It's still a big unknown as to why these things exist, why the cyanobacteria produce them, why certain blooms produce them over others. And we know that based on some studies that both cells and their toxins and metabolites can be transported from where bloom density is off-site. We know this from a Kansas River System study that was performed, I believe it was in 2016, where we saw that toxins could be transported hundreds of miles downstream. But we also know it from more recent studies like Lake Okeechobee, where both cells and toxins were released to the Gulf and to the Atlantic Ocean. Next slide. So where that kind of leaves us is that, based on these studies, we don't really know what to expect for New Jersey, because other states and other systems waters are not New Jersey waters, and those conditions might be applicable, might not be applicable. So what we're currently doing is there's an ongoing study with the USGS, as well as several other collaborators, Montclair State University, New Jersey Water Supply Authority, and NJDEP, to look at the downstream production, the persistence, and potential transport of cyanotoxins, cyanobacteria, and in the Raritan Basin water supply complex. Next slide. So I had mentioned this is a joint project. It's got a lot of moving parts, a lot of hands that are working with this project. It's primarily being driven by USGS and New Jersey Water Supply Authority. We're working on this as well, doing the toxin analysis, doing nutrients. And we're concentrating this focus on the Raritan Basin water supply complex. And we know that the problem isn't unique to the Raritan Basin supply complex. We find cyanotoxins and blooms in other areas of the state. And one of the things that we do know is that, we find other toxins. Microcystin is the most frequently detected toxin. And as Steve had mentioned, it can persist for quite some time. We just don't know at what rate we expect to see degradation. So if you get a bloom in, let's say, Bud Lake, and it transports down, how far does the toxin transport downstream before you start to get natural degradation from oxidative stress or from natural bacterial degradation or fungal degradation? We don't know. We have some lab studies which could suggest something, but actual actual field data to support it is very limited. So we know that in the state that we're getting increased frequency and duration of blooms. And we in the Raritan Basin supply complex, we've found toxins of concern like anatoxin A and saxitoxin that have been found in drinking water reservoirs. So it is concerning, and this is something that we're trying to understand and study. So what the study design basically is looking at is it's looking at both transportation of cells and toxin, and what's their ultimate transportation and fate downstream to drinking water intakes. So while doing this, we're trying to evaluate the real-time conditions to see if we can figure out, when a bloom might make that switch. Is it temperature driven? Is it, linked to rainfall? Is there a nutrient spike? Is there something? We're trying to see if we can tease out in New Jersey, what's the switch between a bloom to a toxigenic bloom. And we're also using these things called Spat Samplers, which are actually pretty interesting. So they are a type of resin that you can embed into, or you can deploy essentially into a water body, and they passively collect toxin as it moves through the water column. So we grab discrete samples, or single point samples. So you get the toxin concentration at that point. You don't know whether it's high or low. It could be really high at that point or really low at that point, because the cells move through the water column. So what a spat sampler does basically is it sits in the water column for a set duration amount of time. And what happens is you go back out and collect it, and then the toxins are absorbed to the resin. You can then elute the toxins off the resin and sample that. So you can get an idea of what's the concentration of toxin over time. So if you do have a bloom that's, let's say, somewhere else in the water body that you didn't sample at, you can figure out, is there a high amount of toxin, low amount of toxin? You can get a better idea spatially of what you're looking at for your toxins. Next slide. So this is kind of an idea what the study area looks like. This is the Raritan Basin water supply complex. You can't read the map. It's, I know, I understand. But basically starting with Bud Lake, which is the headwaters of this, we basically have a number of set points that we're measuring throughout to measure, where and if toxins are moving, toxins in cells. Next slide. So what we have so far basically is it's still really preliminary. The study is still ongoing. This is the second year of the study. So what we're finding so far generally right now is the headwaters have about a 20 times higher cell count or cell density than Spruce Run Reservoir. Spruce Run does have an ongoing bloom. We basically are finding, generally speaking, we're seeing that Bud Lake tends to be higher than Spruce Run, which is then higher than like Rosedale Lake, and these are all connected. We are finding toxins, which is not terribly surprising, but we're not finding them in really high concentrations. But again, this is all still preliminary. And what we're finding in terms of the molecular work is that we're finding the genes to make these toxins, so that potential to make these toxins is there. But again, we don't know all the factors that control toxin production. So the presence of the gene doesn't mean the toxin is going to be there or that the bloom will make toxin. It just means that the potential for the bloom to make toxin is there. And until we can better understand the switch that causes the cells to start making it, it'll always just be a potential. Next slide. And this is basically the contact information and just the references. And I believe that is it. Thank you.
Thank you so much, Rob. That was really pretty fascinating. I'm going to now invite Gabe Mahon from the Bureau of Stormwater Permitting and Water Quality Management to talk to us about HAB mitigation techniques.
Hi, everyone. My name is Gabriel Mahon, and as was said, I'm the Bureau Chief of the Bureau of Najipi Stormwater Permitting and Water Quality Management with the DEP. For any of those who aren't familiar with that name of the Bureau, that's because we recently underwent some reorganization. So it was previously the Bureau of Non-Point Pollution Control that managed a lot of the same stuff. So maybe you're more familiar with that name. Go ahead on to the next slide. So there's really a few ways I'm going to talk about helping to prevent HABs, but basically it all circles around keeping nutrients from getting into the water in the first place. So there's a couple of ways we can do that. One is to, and the easiest and probably the best one to target, is to reduce or eliminate nutrients at the source. Okay, so obviously if there are no nutrients at the source of the stormwater runoff, there's no nutrients for the stormwater to pick up and to carry into the water, and thus no nutrients get into the water. The next one is to prevent the stormwater runoff from reaching the water. Okay, so even if you couldn't eliminate the nutrients, at least maybe you can eliminate the stormwater from getting to the water by infiltrating it or something like that, or letting it get absorbed by vegetation so that it doesn't actually make it to the water. And in that case, it can't carry the pollutants into the water. If you can't do that, then the last option is to take that water that you are discharging into the receiving water body and to treat that, ideally through some sort of green infrastructure. And last thing I'll touch on is managing septic systems properly. Go on to the next slide. So first, reducing or eliminating nutrients at the source. There are a lot of different techniques on here. A lot of them, though, as I go through them, I'll be mentioning that they are requirements under our MS4 permit. MS4, for anyone who's not familiar, stands for Municipal Separate Storm Sewer System. And we issue those permits to any municipalities or highway agencies or public complexes that operate their own storm sewer system, and they discharge those into waters of the state. So the first one on here is to minimize the use of fertilizers. Of course, fertilizers are essentially just nutrients you're putting down onto your lawn and plants. So any runoff that carries those into the river or lake is going to bring nutrients with it. Now, in New Jersey, there is a fertilizer law that requires that some percentage of the fertilizer be slow release and not water soluble. So that definitely has helped in that case already. But we have to make sure that we are following that law, and then in the case of the municipality, enforcing that law, because it is an MS4 permit requirement that they enforce that law. And just keep in mind, if you're a resident, you're applying fertilizer, it's not necessarily the worst thing in the world, but definitely don't apply your fertilizer right before it's going to rain, because it's going to be not effective for you and it's going to get carried off into waters. Next thing I want to talk about is picking up pet waste. And from the municipal side, enforcing the pet waste ordinance that's a requirement of the MS4 permitting program. So obviously pet waste is a source of nutrients. And if you just leave it out on the lawn, it can be exposed to stormwater, and those nutrients can be carried into the storm drains and then eventually into our streams and rivers. The municipality is required to have an ordinance that prohibits leaving pet waste out in any public areas. So for example, if you're at the park, the municipality probably has an ordinance that requires you to pick up your pet waste if you're at the park. That ordinance is not going to apply to your own backyard, but keep in mind that your backyard can still be a source of nutrients if runoff from your property makes it into a storm drain or makes it directly into a river or a lake. Next thing to talk about is wildlife feeding. So again, MS4 permit requirement that there's going to be an ordinance that you can't feed wildlife in public areas. Again, nothing would prevent you from doing this in your backyard, but really, if you're along a stream or river, the results are the same. The major, as we already heard, the major wildlife source you want to think about here is probably geese. For a lot of reasons, we already heard that geese produce a lot of nutrients, but also obviously, as we know, geese tend to congregate along waters, especially ones where it's just open grass areas where there's not a lot of vegetation to absorb their nutrients. On top of that, you may not know that New Jersey is one of the highest density states for geese in the country as well. So we do have a tremendous amount of geese here. And so just keep that in mind, that if you're feeding wildlife, it's going to attract them to come back there more and more, and it's just going to produce more and more nutrients that are a problem for HABs. So definitely something you want to avoid. And if you're a municipal official, or if it's your own private property where you own a lake or a river, consider installing measures to reduce geese there. Vegetative buffers are a good way to keep them out, but also fencing or mylar barriers also can help to reduce geese. We have a lengthy list of wildlife management techniques in our MS4 permit guidance that you can consult, and a big percentage that is dedicated to geese. Next thing is to properly manage your yard waste. So leaves, grass clippings, weeds that you pull, all that breaks down into nutrients that can be carried by stormwater runoff into our lakes and rivers. So you want to avoid that. And again, the municipality has a yard waste collection ordinance requirement as part of our MS4 permitting program, and that's going to require that they don't put the loose leaves or debris within 10 feet of any storm drain, and that they only put it out during the collection cycle that is allowed by the municipality. And so if you're a resident, you definitely want to follow that, but if you're the municipality, you also want to make sure you're enforcing that, right? If people are putting out their leaves 2 weeks before the collection period, make sure you knock on their door or send them a notice of violation or whatever you call it in your municipality to let them know that's not acceptable. Or if they're putting them right along storm drains when the requirement is to be at least 10 feet away. And then for municipalities, the next thing to talk about is street sweeping. Obviously, street sweeping picks up pollutants from the street. So anything that you pick up from the street doesn't wind up in the stormwater runoff from the street and doesn't wind up in rivers. So any amount of street sweeping is a good idea. The MS4 permit does have a requirement for street sweeping, but it is limited only to the commercial areas of your municipality. So it's really a good idea if you're in an area that's dealing with HABs to consider going beyond the minimum requirements for our street sweeping and start sweeping in those residential areas as well, because again, you're removing those pollutants directly from the source. Another good thing to do is to retrofit your storm drains. The permit does require storm drains to be retrofitted when you're repaving the roadway, but there could still be storm drains in your municipality that haven't been retrofitted. And basically an unretrofitted storm drain has a much bigger opening and will allow a lot more debris to pass through and thus make its way down into your discharge. So it's a really good idea to retrofit those, and can easily be done by just screwing or welding like a bar or a plate across the front of a lot of inlets. And the last thing here to reduce or eliminate nutrients at the source is to really educate your residents and your municipal staff about the reasons behind all this, the effects it has on your community, right? If you have a HAB in your community and it closes your lake down, that's going to have tremendous effects on your community as a whole. So everyone should really have an understanding of what they're doing. Go on to the next slide. So if you aren't able to eliminate the nutrients, then you want to prevent the stormwater runoff from discharging. So this is going to include planting buffers of native vegetation around your streams and lakes so that the water can flow through there and be absorbed and hopefully not discharged, or at least the nutrients can be taken up by the plants instead of making it into the discharge. Otherwise, you're going to want to install green infrastructure such as rain gardens. This can do two things. First of all, a lot of green infrastructure facilities, if the soils allow for it, are going to infiltrate stormwater rather than discharging it. So then it's not going to carry the pollutants. Or these systems can also actually reduce pollutant loads of nutrients as well. On the municipal level, ensure that developments are complying with the stormwater rules. So major developments need to comply with the residential site improvement standards, which basically require them to meet the stormwater rules. And major non-residential developments have to meet the local stormwater control ordinance, which has to be at least as stringent as the stormwater management rules. We've done audits of a lot of municipalities, and let me say that I think there's room for improvement here in municipal reviews. So, if you're a municipality, make sure you're doing a good job here because this has an effect on the quality of water leaving these sites. And another big topic is the BMPs at the municipal maintenance yard. Make sure your municipal maintenance yard is properly managed. In particular, your compost operation, if you have one. Also, other important areas for HABs are the place you deposit your street sweepings, the place where you deposit the debris you collect from your inlets and catch basins, and any other materials that are going to break down and wind up, providing nutrients to these waters. I particularly say compost operations because our permit covers compost operations if they don't discharge to surface water. The MS4 permit covers them. But if they do discharge to surface water, that's actually not covered by the MS4 permit, and you really do need another permit to cover that. And from what we've seen, a vast majority of the compost operations actually are unable to completely contain their discharges on site, and they wind up with discharges to surface water. So keep that in mind in the way you're managing it. That's going to result in nutrient-laden water being discharged off your site. And if it's going to a lake that has HABs, that could be a big contributor. Go to the next slide. These are some typical nitrogen phosphorus removal rates for BMPs. Now, these are really averages. It's going to be very dependent on the site and the BMP that you install. But the main thing I wanted to convey with this is you look at what BMPs are: green infrastructure, bioretention basins, infiltration basins, pervious paving systems. You're going to see that they have the highest removal rates for phosphorus, nitrogen. But if you look at, say, like a regular extended detention basin, which wouldn't be considered green infrastructure, that's down on the lowest end of what would provide, or what nutrient removal rate you're going to see from that BMP. So keep in mind that you really want to target green infrastructure systems with this. Go to the next slide. You need to have a stormwater outfall maintenance and inspection program. And this works whether it's municipal or it's a private development. You want to inspect your outfalls when it's not raining during dry weather for illicit connections. These could be sewage, which obviously is a huge input of nutrients if you have sewage directly connected in there. Could be industrial discharges, wouldn't have anything. Could be drinking water, which actually could contain nutrients. And it could even be groundwater, which probably isn't normally a problem as a source of pollutants. But if you're in an area where there's all septic systems and the septic systems aren't necessarily functioning the best, the groundwater could also be a source of nutrients. Erosion and scour control, of course, if the area around your outfall is eroding, you want to prevent that because the soils that are eroding could be a contributor, and trash and other debris around your outfall. You want to clean that up, prevent it from breaking down, and prevent it from looking terrible at your outfall. Go to the next slide. And then there's the stormwater facility cleaning and maintenance. Again, this is both at the municipal level, being a requirement of the MS4 permit, but also if you're a private owner of something, you want to make sure it's maintained. You've got your BMPs, detention basins, infiltration basins, manufactured treatment devices, your green infrastructure. Most of these are going to contain some sort of vegetation. So you want to remove that vegetation, whether it's grass clippings or it's weeds that you remove from the system. You want to remove that from the basin entirely and instead of letting it break down there and just have the nutrients just go back into the system. You remove them out, bring that to your properly managed compost operation and get them out of the system. Inlets, you need to inspect those regularly for debris. In my opinion, it's best to inspect those when debris is likely, right? So that's when leaves are falling off trees or after heavy rains. And then your catch basins, you need to clean out those catch basins on a regular basis. A study from EPA shows that after about 40% of the catch basin is full, it really starts to lose its water quality benefits. So you really want to clean those out before it reaches that 40% capacity. Go to the next slide. All right, and the last thing I want to touch on briefly is septic systems. So you really want to avoid introducing not just phosphorus, but all nutrients into your septic system. Particularly to prevent phosphorus, you want to not use detergents and soaps that have phosphate. And that's a very common ingredient. But there are plenty of alternatives that don't contain phosphates available. Eliminate the use of a garbage disposal. Aside from being a source of nutrients going into your system, also a garbage disposal is not necessarily the greatest idea in a septic system. It can work out, but also it can contribute to the system being clogged up with particles that aren't broken down enough. So it can definitely lead to your system not functioning at its peak down the line, and any lack of function is going to be contributing potentially nutrients downstream from your system. Again, also using water efficiently anytime you have a septic system for the same reasons. You want to ensure your system is properly maintained, so it's going to need to be inspected regularly for any problems. You want to have it maintained and pumped out regularly. We recommend that you do that on at least a 3 to 5 year cycle. If you have an effluent filter, you want to replace it or rinse that out regularly. and definitely immediately repair any malfunctioning system. There is also the possibility of installing advanced wastewater treatment, particularly for systems that are in areas of high groundwater where you can't get the kind of treatment you really want the system to be providing. Now that can be costly, so maybe not an option for homeowners, but more of an option for larger facilities like churches and restaurants, and maybe the municipal building if it's on a septic system, all sorts of larger buildings like that. Advanced wastewater treatment can really eliminate the source of nutrients from a septic system. And then the last thing is to consider extending sewer lines where it's feasible. If you're in areas around lakes, you're often going to be dealing with high groundwater where septic systems really are not going to work the way they will in areas where they have a lot more separation from the groundwater. So you're not going to be getting that phosphorus removal in the soil that you want. And in those situations, there may be little you could do other than having large mounded systems. So sewer lines might be the best option in some of those cases. All right, and that's it for me.
Thank you so much, Gabe, for this overview of best practices. Very thorough and a lot of information. I'm going to now invite Dr. Steven Tuorto from the Watershed Institute. Steve's the Director of Science and Stewardship there, and he's going to talk to you about some of the work he's doing with emerging practices to mitigate HABs.
Thank you, Marjorie. We can jump right to the next slide, I think. Tried to keep things pretty simple and to this project and visual just for time's sake, and we can do questions at the end, obviously. So, the overall project, there are a lot of tools. Gabe went over a bunch, mentioned some of the green infrastructure practices that are going in. But there are, of course, with the emerging problem of HABs, we have emerging technologies that are being tested and being more scrutinized for use in mitigation. So what happens after the nutrients hit, or what happens after we have warmer climate, that kind of thing. So just to put the project in perspective, it's part of a partnership with Mercer County Parks Commission. When DEP's Watershed Restoration Department program came out with an RFP, a call for proposals on work to try and test mitigating HABs, we decided to work together on Rosedale Lake. Rosedale Lake was mentioned by Rob earlier, and has been one of the lakes that has a historical record of HABs over the last 5 or more years. Just for perspective, you see on the map there in red where Princeton and Lawrence Township is, where the Watershed Institute Reserve is, and in the purple circle there is where Rosedale Lake is. It's pretty high up and a tributary to the Stony Brook watershed, which feeds into the Millstone just above Carnegie Lake. And you see the picture there that I said historically having HABs, the picture in the lower right I took a few weeks ago after we deployed our wetlands. And you see we had a pretty strong bloom in swing there. Next. So there's a lot to talk about, but I'll just try to stick to the basics in terms of using constructed floating wetlands as a tool. Essentially we have a wetland, right? Wetland serve a very important function ecologically, but also in reduction of pollutants and trapping of carbon and all types of organic matter, but in sort of a limited capacity. And a lot of that has to do with the fact that surface area is limited, and so is oxygen. So you see in the left-hand depiction from a study from Luck et al., just depicting that what a floating wetland is you have a synthetic base that you create and you make to be buoyant, but that you can then plant native wetland plants in, so that their root structure can grow through the bottom of the island and into the water column. So what we have, first and foremost there is we remove that oxygen depletion part in a native natural wetland. And we get more flow through and around the roots and even through the material that the floating wetland is made of. And on the right-hand picture, courtesy of Floating Island International, shows a little bit closer up close and personal look at the root structure and even the matrix that makes up the island base, the bed of the floating bed. It's made from a filter material, which I'll show in a minute. But what we get is a very highly increased surface area to volume ratio for things like root formation and growth, and exposure to the water that's full of these nutrients, but also the microbes, and maybe even more importantly, the microbes that are associated with these roots and in the filter material itself. So we get a high rate of removal with a constant sort of flushing past the materials that are doing the removing, or the organics that are doing the removing, of all types of things, including some more toxic things. But of course, the focus here are the nutrients such as nitrogen and phosphorus. There is quite a bit of research out there now, looking at things from various perspectives on the efficacy of using these things. But if I pull from a few of them and either do some calculations with that myself, or pull from others some of their calculations, you get things like estimates of a 250 square foot constructed floating wetland in terms of surface area to provide root surface area, and to provide growth and colonization space for microbial activity, is about equivalent to 170 times that of a natural wetland. And I think that's compared to 1 acre of wetland. So 250 square feet compared to 1 acre, and the potential to mitigate approximately 11,000 pounds of algal biomass with the same size floating wetland, 250 square feet. Next. So the design, in a nutshell, the project actually is employing 3 strategies: barley bale deployment, as well as aeration devices. But of course, we're talking here about the floating wetlands. And so often we're thinking along the lines of how much floating wetland does it take to really make a difference? And there are some numbers thrown out there. So how, square foot of a floating wetland device or treatment surface per acre, or volume of lake. And what I really found interesting in my own review was this study done by Luck et al., which was actually a review of all of the relatively easy to compare studies on the efficacy of floating treatment wetlands on reduction of algal blooms and nutrients. And they found that really one of the key factors was not so much the percent area coverage that you got, so square foot of island to square footage or acreage of water body, but was maybe more important that the study design didn't allow for what they were calling short circuiting of the system. So short circuiting of the filter. If you put this out there as a filter, but the water that needs to be filtered is making its way around it, either vertically or horizontally, then you have a short circuit, and it's going to be a lot less effective. So in the bottom left-hand picture, we see our original proposed deployment for floating wetlands. And that was identified as two marked and 1 unmarked tributary into the island, because it appears that relatively little runoff is coming directly from the surface within that sort of watershed. Moving on, after funding, we did preliminary surveys and more research and design, and delineated the watershed of each of the tributaries. Found that the contribution to the smaller tributary was pretty minor, and also well forested. Again, that the runoff from directly around the lake was sort of minimal, even though there is wildlife there, and so that we could probably stick to treating the 2 main tributaries, which involved runoff from a lot of ag land and some developments with detention basins. And then we also ran some drone surveys after rain events, so we could track sediment plumes from the tributaries, so that we could be sure we were kind of intercepting the flow, and to make sure we didn't have horizontal short circuiting of our plan here. We went out and ran depth profiles. Don't judge how straight the profile lines are. It was a windy day, but we ultimately determined, noted by the green lines there, that we would be okay to sort of put a stretch of islands right across at those locations. Next. So next is the design. We've ordered commercially available. There are a few There are a few companies that do this for a living. They're rather expensive, but we also have, over the last 5 or 6 years, done some DIY builds comparing to the commercial ones that we bought, and have kind of stuck to building our own. And so the basic design in the upper left-hand corner there, the schematic showing, we use a polyflow non-woven polyethylene. It's basically the same thing that, filter material that you would buy for your water feature in your yard, or even your filter in your fish tank, just thicker and in very, very large rolls. You cut them to the length you desire. We have we put them together in 4 layers of the white, a little bit less dense for surface area infiltration, lower layers, and then we put a black layer of the slightly more dense and more UV resistant black layer on top, just to take some of the weathering. We use a 2-part expanding aeromarine foam, closed cell foam, that it's a urethane base that's able to pour straight through before it starts to cure the filter material, and then it expands, glues the whole island together and makes it float. And then I have a series of photos just sort of showing the build. We start with some bottom layers, creating some buoyant pods on them, and then going to the right, you just see the layout there. Lots of filter cutting and pouring. Then we glue the top layers together by pouring more foam through. If you go up to the top picture there, you see us. Then you drill your plugs out. You take plugs out for planting your plants. We also insert in the corner and at each midway point PVC tie rods glued also in place by the aerofoam. And finally, we cover it with a core jute matting just for some initial integrity before the roots start to take over and hold things together, and for like a substrate layer. Next. And then there's the deployment. We transport the islands to the location. We had a whole bunch of staff out for one of our first in-person events since restrictions started lifting a little bit. So that was fun. And plant the islands, help haul them out to the boats, tie them together. We used primarily Juncus and Carex, so bulrush and soft rush type native grasses, and then just for ecological and floral aesthetics, milkweed and cardinal flower as well. Tie them together, strap them to the boat and pull them out. Deploy them. We had pre-deployed anchors, tied them together and tied them to the anchors. And you see in the lower right-hand corner, achieve sort of that whole stretch that'll intercept the majority of the inflow from one of the tributaries. Next. And now we watch and wait. There's wide variation of these being tested, and the technology itself is actually an ancient one, right? It's basically floating synthetic based, or at least, in ancient times it probably wasn't, but it's hydroponics, where people were growing plants for food and ancient civilizations out on the water, but being revived for a new purpose. It's never set it and forget it. You always have to monitor these things and have some maintenance plans. We had, you plan for a late April, early May deployment because the weather should be decent and give the plants a chance to sort of establish themselves to be able to handle some heat and dry times. But instead, in May, we had a heat wave and a drought, and the water was wicking out of the filter material faster than it could, or evaporating out faster than it could wick up. So they were sitting completely on top of the water in a heat wave. And were baking. So we had to go out and add some weight so that the plants could recover and establish. And now the islands are sitting right where we want them. And you see in the bottom hand picture, that's a picture just a couple weeks after that they're doing really, really well. We are monitoring and haven't seen any wildlife approaching the islands yet, but are going out anyway to deploy goose netting. I stole some pictures actually from Dr. Souza's, one of his presentations, just to show that if you don't protect your features from wildlife, you see that bottom middle picture that they just get destroyed and then are worthless, obviously. And then we're monitoring. The day we deployed pre-deployed the anchors, I could see in the water that things were building up to a bloom. And I said, hopefully it holds out a little bit. But by the next day, we went to deploy and plant. And sure enough, that top left-hand picture shows the paint slick there associated with a bloom. But we'll be out. We are one of those organizations that's partnering with DEP and EPA to begin some citizen science monitoring of phycocyanin with phycocyanin meters. We'll be monitoring the root growth, and of course, monitoring the data that DEP collects with their bottom left hand. And I think that's it. Thanks for listening.
Thank you so much, Steve. That was great. I'm going to now invite Dr. Rob Newby back to turn on his camera and talk to us about resources to support municipalities in addressing and managing harmful algal blooms.
Thank you kindly. All right, next slide. So myself and Vic Poretti, we were invited to be part of the Interstate Technology Regulatory Council, or ITRC, technical document on harmful cyanobacteria blooms, or HCBs, and which was comprised of both stakeholders, academics, industry experts, state and federal employees. It was about a 2-year draft process. And it really comprised a number of really key modules, which I think are really interesting to a number of people who are interested in understanding all of the factors in response to a bloom. So it's been published. It's available freely at the link. So if someone can publish that in the chat, I'd greatly appreciate that. And there'll be more training that's available at the end of the year. And at the end of the slide, there's a link to the actual date and registration. Next slide. So one of the key modules, which I think is really important, is there's a nutrient reduction and control module. So kind of an overview of what a watershed actually looks like and all the potential points of input. So many people get hyper focused looking specifically at the bloom in that water body. And that's critical. I mean, it is important to understand that, but there's also kind of inputs that could be coming in from far upstream that you don't quite understand. So what the tool does is allows you to click on each individual point. So next slide, by hovering over an area, it tells you a bit about what that potential input is and tells you why and what potential steps could be done to reduce that input. Next slide. Followed then by there's a management module, and this is actually really important. So Dr. Souza had mentioned before that copper sulfate is one of the immediate go-tos, is what people think of immediately to treat a problem. But copper is, kind of a double-edged sword. You can apply it and you can treat your problem, but there'll be rebound. And we have an abundance of documentation of rebound effects. So if you're looking at sort of novel or new techniques, or some things that have been tried and true in the literature, you can use the management module, which is developed looking at both kind of an intervention and prevention. So there's 2 types of techniques: things that can prevent the bloom from occurring, or things that you can do in the immediate, as well as different levels of technology, whether it's an emerging technique, if there's a little bit of limited field data, or the substantial data. Followed then by your water body size. If you click on next, give you an idea as to what kind of what you can expect. Next slide. So if you're looking for an intervention technique that has substantial data on a lake or reservoir that has small surface area, so on and so forth, it gives you this list. And if you click on the next slide, it pops up with the fact sheet and tells you a bit about what the technology is, limitations, where it's best approved. And what it's supposed to do is help guide your conversations with a certified lake manager. So if you are looking at how to treat your problem, these will give you some ideas as to where to go. Next slide. There's also a cost table. So what other lakes have experienced, a cost per acre, and the source. So you can see, kind of where this has been used before, and how much is cost per acre and when. So you can get an idea as to, how much in 2021 dollars it's going to cost. Next slide. And then it's blurry, I'm sorry. If you go to the website, there is a monitoring tool. And this kind of goes into a question that was in the chat before of how you can best monitor blooms. And there are some low cost tools that are presented here that are really simple that you can do to see, if it's a cyanobacteria bloom, or if you have a little bit of money to spend, some low cost things that you can buy to help you with monitoring. But there's also the free Fico sign-in handheld meter program, which Vic had previously presented. Next slide. So there's the link. And then there's the link for the training, and then my contact information and Vic's contact information. So the website has more than just the modules for training. It's really in depth. It's basically the current state of the science for all things HABs, or all things cyanobacteria HABs. Right? Thank you.
That was very speedy, Rob. Thank you. So on cleanup, but not least, is Eugene Chebra, who's going to speak to us about some of the financial resources. We've been getting a lot of questions about that, so maybe some of your questions will be answered. So Gene, take it away.
Thank you, Marjorie, and thank you for having me. And I hope to be able to answer some of the questions that I've been seeing in the chat. My name is Eugene Chebra, Assistant Director in the Division of Water Quality, and our group administers the Clean Water and Drinking Water State Revolving Loan Programs. Next slide, please. Collectively known as the Water Bank. The objective of the Water Bank is to provide low-cost financing. We cover planning, design, and construction costs. We like to pride ourselves on being pretty innovative to be able to fund any project that has any nexus to protecting, maintaining, or improving water quality. So, like I said, we like to be innovative to make any nexus to projects that meet that criteria, fund everything from equipment purchases like street sweepers, jet vac trucks, weed harvesters, up to multi-MGD treatment plants. Next slide, please. The source of funding, it's a combination of funding sources. We get our main funding source from EPA and a capitalization grant. It is provided by a state match. We have to match those grants. And then we further leverage those funds with our financing partner, the Infrastructure Bank, formerly known as the New Jersey Water Infrastructure Financing Program. And we get repayments from projects that we have funded through the years. We have quite a bit of experience. We have 30 years of experience. Next slide, please. We've issued over 8.7 billion in low interest loans. This year, we may have our largest year on record. We have 2 more weeks left in the fiscal year, but it looks like we will be closing on approximately 700 million in low interest loans this fiscal year, one of our largest years on record. Next slide, please. The structure of the loan, up to 30-year term loans. We have rolling applications. There are no deadlines. We have rolling awards. We award based upon a readiness to proceed. The drinking water side of the program, which we're not going to focus on today with the HAB presentation, but we are running low on drinking water funds. So those loan awards are based on priority ranking. We issue bridge loans, construction loans, which are similar to bond anticipation notes. So that at the end of the day, when you long-term finance, you finance to the exact dollar that you need. And there are no out-of-the-pocket expenses. I take pride in a project that we did with the Bayshore Regional Sewage Authority. It was a large project, and they did not have to come out of pocket a single penny. They would get the bill from the contractor, send it to our program. We would process that payment in time for them to pay the contractor before within the pay period. Next slide, please. Everything in the program is online, project submission, priority ranking, letter of intent, all these can be submitted online. We will provide links to our intended use plan on how to submit your documentation for financing. Next slide. We have made a change in the years after the ARA years that we award based upon the construction of a contract. So years prior, we could award loan funds based upon an engineer's estimate. This proved to be problematic in that we had a lot of unspent proceeds. The project would either never go out to bid or would be delayed. So now we certify a project for financing after the project has gone out for bid and award is recommended. And like I said, it's a short-term loan award that will be taken out with long-term financing. The iBank just sold bonds for their piece last month, and they got a very favorable interest rate, 1.88%. So when you combine it with the DEP zero interest loans, our loans are currently being made at under 1% interest rate. So very favorable terms. Next slide, please. Like I had mentioned, the online system is called H2 Loans. Everything is done online. You set up an account with our iBank, the sister agency, our iBank. Information uploaded is only for your eyes and our staff eyes. No other applicant can see your information, but all information is loaded up online so that you can see the status of your project and we can see the documents that need to be reviewed. Next slide, please. So this is our mechanism to do our call for projects, the intended use plan. It's an EPA requirement. It sets out the terms and conditions that we're offering. For the department, there is no higher priority than HAB abatement projects and combined sewer overflow abatement projects, projects that will eliminate the discharge of raw sewage during storm events. Next slide, please. So within that intended use plan, like I said, there's this category called water quality restoration. These are for capital improvement projects and equipment purchases to eliminate or reduce HABs. I'll get into a little bit on the type of technologies that we can fund. We have not funded a floating wetland as of yet, but that would be like an eligible project. Next slide, please. So the $10 million of principal forgiveness, I can't call it a grant. If I call it a grant, it triggers many other EPA requirements. So it's every time I mention principal forgiveness, think grant, but it is what it is. It's the forgiveness of the principal part of your loan. So the department for next fiscal year, beginning July 1 of 2021, running through June 30th of 2022, we've set aside $10 million in principal forgiveness for HAB-related projects. There's a $2 million cap so that not one entity could come in and eat up all of the money. So at 50% principal forgiveness at the $2 million PF cap, it equates to a $4 million project. So 50% of that project would be principally forgiven. 25% would be a DEP interest-free loan, that's at 0% interest. And 25% would be our sister agency, our financing authority, iBank, which is market rate. And like I mentioned, they had recently sold bonds at 1.88%. So pretty favorable terms and conditions. For projects over 10 million, the rate above 10 million is 75% DEP at 0 interest rate, 25% iBank, and up to 10 million. Costs over 10 million would be 50% DEP and 50% iBank. Very high cost projects, but we really don't expect to see that in the HAB area. Next slide, please. Like I mentioned, these are just some of the examples of the type of projects that we finance. Our bread and butter are wastewater treatment plants and collection systems and pump stations, but we also finance non-point source, stormwater management, site remediation, water meters. Like I mentioned, equipment purchases. We funded an interesting project along the coast where 3 towns had coordinated to purchase street sweepers, and they share that equipment between the 3 towns along the coast. Next slide. Like I mentioned, stormwater management. Within the CSO NHAB category, like Gabe had said, we are really encouraging green infrastructure. Green infrastructure picks up the phosphorus and nutrients from the system. So we really encourage the use of green infrastructure. Downside of the program, this may answer a couple of the questions. We cannot loan funds to individuals or homeowners associations at present. We are investigating being able to loan funds to local financial institutions, local banks, to make those funds available to homeowners and homeowners associations. But because of the structure of our program with using the iBank and going to the market to sell revenue bonds, at present, eligible recipients of financing have to be government entities, either a township, a county, or a municipal utility or sewage authority. And we can finance individuals or homeowners associations using those eligible entities as a conduit. For example, if a town wanted to put in a cluster septic system to take several homes offline, we could finance that project through an eligible entity. But right now, the downside of the program is that eligible entities are limited to governmental agencies. Next slide. Oh, and that is it. There is a link to our program, DEP, Division of Water Quality, Municipal Finance and Construction element. That's where you can find all the documentation on our intended use plan, the packages that we are offering. I would say keep your ear to the rail. We may be issuing an amendment to the IUP should American Recovery Plan funds become available. And there is also a bill before the, I believe it's up before the Assembly Appropriations Committee today, A5778. It's also S3618. And that bill appropriates $10 million for lakes management. So that's another bill to keep a close eye on to see how that progresses, and keep your ear to the rail for additional financing opportunities.
Thank you so much to all our speakers. Please turn your cameras on. We ran way over, but I guess we were super ambitious with so many of you. But we've been monitoring the chat box, and I think Carrie's going to try to pull out maybe one question that you all were pretty good at answering questions in the chat box, but you also, you can go to our website and you can see where everybody's located, and you can send us notes as well, and we'll pass them along to the speakers. So go ahead, Kara, if you can grab one.
Great. Thank you, Marjorie. And thank you all to the speakers for such a wonderful presentation. Okay, so as an education person myself, I'm going to choose this one kind of selfishly. You have some wonderful tools and some wonderful resources out there. There was a question asked about, how we can best educate residents and municipalities in lake communities, and if there's been examples that you've seen that have worked really well in the state of New Jersey or outside of it. I'll pose that to anybody who wants to chime in there.
Well, I'll jump in. I mean, I don't know about anything specific to lake communities, but when it comes to stormwater in general, I think what you really need to do to educate residents is you have to get across to them the impact that it has not only on their property, but how that impacts other properties and how that impacts the whole community. And I did see one question in the chat, like, how do you get to people that really don't care about the community? Well, I guess if you're living at a lake, the way that you get to them is obviously they're at the lake because they want to recreate in the lake in some way. And you're going to have to point out to them that, listen, if it's shut down by a HAB, you can't go swimming, you can't go fishing, you can't do all the things that you live at this lake to be able to do. So it's really showing them the benefits to their community and also the effects on themselves personally.
Yeah, and Carrie, I'll add to that as well. I mean, there's some great resources out there. The North American Lake Management Society, I was a former president of NOMS, the New Jersey Coalition of Lake Associations, and the New York Federation of Lake Associations have, there's great sources of information for folks that live on or around lakes, to answer a lot of their questions. And put them in direct contact with experts that deal with different types of management strategies. But the thing is, and everybody that's on the webinar today can tell you that that every lake is different, and you need data, and that data needs to be sorted out to identify what's relevant. But I mean, one thing that we do know, and everybody has stressed this, that phosphorus is the key contributing problem. And whether it's stormwater management, it's septic management, it's some type of internal phosphorus control, that needs to be done if we're going to battle HABs successfully. Less food means less HABs.
Thank you. Thank you all so much. On behalf of us, we're just very grateful that you participated. What a dream team. And thank you again. I just want to let you all know that we'll be posting a recording of this, and we will be sharing all the questions that you provided in the chat with all of our participants today, all of our presenters. So your thoughts are very welcome. If the next and last session in this series is on June 24th, we're going to be talking about how much did climate change contribute to the damage inflicted by Superstorm Sandy. There'll be folks from Climate Central, Rutgers and Steven, to answer the question. It's a new study that they released. So we look forward to having you join us then. And that concludes today's program.